Okay, this is the back end of chapter 25. We were discussing the urinary system, and what we want to do is continue our look at the nephron and how it does its work. So, when we're talking about reabsorption, remember we're talking about taking stuff out of the filtrate, which is the liquid that's in the nephron, in the tubule, and putting those contents back into the blood. Most of the tubular contents are reabsorbed into the blood through selective transepithelial processes. As an example, all organic nutrients are reabsorbed, and water and ion reabsorption is hormonally regulated and adjusted. This includes active and passive tubular reabsorption. And there's two routes that this can follow. It can be either transcellular or paracellular. Transcellular means through the cells, paracellular means around the cells. And what we mean by active or passive reabsorption, active involves the use of ATP as an energy source to drive solutes against their concentration gradient, while passive tubular reabsorption requires no input of energy and is driven strictly by the concentration gradient of the substance in question. So in the transcellular route, we have an apical membrane of a tubule cell and the cytosol of a tubule cell. And so what happens is that the substance to be transported moves through the apical membrane into the cytosol, through the basolateral membrane of the tubule cell, and then through the endothelium of the peritubular capillaries and back into the blood. In the paracellular route, we run between tubule cells. This access is limited by tight junctions, but it's leaky in the proximal nephron. Examples of substances that can be moved by this route include water, calcium, magnesium, potassium, some sodium, primarily in the proximal convoluted tubule. Remember when we talked about the four parts of the nephron, the Bowman's capsule, the PCT, the loop of Henle, and the DCT, different activities are occurring in different parts of the tubule, and the reason for that is that the tubule is not one uniform structure made up of the same tissue throughout its length. There are different pumps, different channels in different parts of the tubule. So here we can see a diagram showing you some of these different transport routes. Okay, this is again available in your book. This is 2513. So refer in your textbook to this figure when we discuss each of these cellular routes in detail. So as an example, let's take sodium. Okay, this is the most abundant cation in the filtrate. It's a major component of membrane potential. It's also a major driver of water movement, because remember that osmosis dictates that water will follow solutes wherever they go. It's transported across the basolateral membrane through a pump called the sodium-potassium ATPase that sends this cation into the peritubular capillaries. The transport across the apical membrane occurs when sodium passes through it by secondary active transport or facilitated diffusion mechanisms. Now, you may have forgotten how these work. Let's talk about facilitated diffusion first. In facilitated diffusion, what happens is that there's a protein carrier that will aid the movement of a particular solute across the membrane, provided it has the proper shape and the proper size. It doesn't just go straight through the membrane. Secondary active transport is a little bit different. Secondary active transport is where the movement of a solute across a cell membrane is actually driven by the gradient for a completely separate solute that's actually established by some form of active transport. Okay, so one of the ways that this happens, for instance, with cells that take up metabolic energy, is that there is a sodium glucose transporter that allows glucose to hitch a ride moving inside the cell along with the transport of sodium inside the cell as it flows down its concentration gradient. So this is one of the ways that we can move a substance through with the aid of a protein carrier, even though we're not directly using the ATP in order to drive the transport. That's why it's called secondary active. Sodium reabsorption by primary active transport provides energy and a means for reabsorbing most other substances, and this creates an electrical gradient that leads to the passive reabsorption of anions, which essentially follow the cations as a result of electrical attraction. Organic nutrients reabsorbed by secondary active transport are co-transported into the cell along with the sodium. And that's what we were talking about when we were saying that the two substances move in together. The gradient that moves the sodium in is actually created by the sodium-potassium ATPase, but it's the glucose-sodium symport that brings both of those components back into the cell. And then, as soon as the sodium enters, it's pumped back out by the pump that's continuously running throughout the process. Examples of substances that move this way include glucose, amino acids, some ions, and certain vitamins. The passive reabsorption of water follows the solutes wherever they go. The movement of sodium and other solutes creates the gradient for water. Water is reabsorbed by osmosis, and it's aided by water-filled pores called aquaporins. And these are special water holes, if you will, that are either introduced or removed from the collecting duct as the body's need to either retain or lose water changes on a moment-to-moment basis. So this is under the control of a very important hormone called ADH. That stands for antidiuretic hormone. And when antidiuretic hormone is secreted, what happens is that you retain a lot of water. And what this means is that the collecting duct is very permeable to water. As a result, as the filtrate moves through it, much of the water in the filtrate, which is eventually going to be the urine, moves back into the kidney and back into the bloodstream. By the same token, if ADH is not being produced, what happens is that the collecting duct is relatively impermeable to water. As a result, the urine is very dilute, and so this reduces blood volume and blood pressure. Aquaporins are always present in the proximal convoluted tubule, and they promote obligatory water reabsorption. They're inserted in the collecting ducts only if ADH is present, and this is what we call facultative water reabsorption. And this again is determined by the body's need to either retain or lose fluid volume. Solute concentration in the filtrate increases as the water is reabsorbed. The concentration gradient for solutes is created as a result. Fat-soluble substances, as well as some ions in urea, follow the water into the peritubular capillaries and down their concentration gradients. Lipid-soluble drugs, environmental pollutants, and other compounds are often difficult to excrete. And what you're looking at here is just a peek at what goes on in the proximal convoluted tubule. This is a nice summary slide. If we were to look very quickly at this and look at each of these phenomena, the numbers here and the explanations match to what's down here in the diagram. So if we look here at what's happening at location number 1, what you're seeing here is the interstitial fluid. This is the filtrate. This is what eventually is going to be the urine. So what's happening here at number 1? Well, the basolateral membrane is that part of the tubule cell that's closest to the blood supply. The sodium is pumped into the interstitial space by the sodium-potassium ATPase, and active sodium transport as a result creates a concentration gradient which drives a number of other processes. Among these, downhill sodium entry at the apical membrane. Okay, so again, this is a result of movement down a concentration gradient from the filtrate into the cytoplasm of the epithelia that lines the tubule. It also drives reabsorption of organic nutrients and certain ions by co-transport at the apical membrane. That's indicated here by 3, and you can see the molecules that follow this symport: glucose, amino acids, ions, and vitamins. The point of this is what? To recover useful solutes in the filtrate, stick them back into the blood so that we don't lose them in the urine stream, because we need to use these either for biosynthetic processes or as a source of energy. At 4, okay, right here, you see reabsorption of water by osmosis through aquaporins. This is an aquaporin, okay, here and here. And what happens here is that water reabsorption increases the concentration of solutes that are left behind. Okay, so as water leaves and solutes remain, the osmolarity of the filtrate goes up. These solutes can then be reabsorbed as they move down their concentration gradients. And then number 5, down here, lipid-soluble substances, as a result of them being lipid-soluble, can easily pass through a membrane, enter the cytoplasm, and then pass directly into the bloodstream. So they diffuse by transcellular routes. And then finally, various ions such as chloride, calcium, and potassium, as well as urea, diffuse by paracellular routes. They go between the cells, right through the tight junctions, and into the bloodstream. Okay, and again, these are flowing down their concentration gradients, And the way that we know that is that there's no pump associated here. Okay, so what you're looking at is down here with this key. You can see a different example of each of these transport processes. So the red arrow, that's primary active transport. That's ATP dependent and relies on a membrane pump. Secondary active transport is not dependent on ATP directly. It is dependent on ATP indirectly. The processes 2 and 3 are examples. And again, they require a membrane channel to move through, a port. And then passive transport is indicated again by the blue arrows here, and that's driven primarily by concentration gradients and not by an input of energy. Okay, so active transport here, secondary active transport, passive transport, all of these going on at the same time. Okay, so if we were to describe the job of the proximal convoluted tubule in a nutshell, it's to recover important solutes, put them back into the bloodstream because the body needs them to do work. Sometimes we can overload the PCT. If we do this, what we do is reach what we call the transport maximum. There's a transcellular transport system specific and limited maximum amount that you can move a solute across a particular cell. If you overload all the pumps and overload all the channels, and there's still solute left behind, that solute's going to stay in the filtrate. The transport maximum is different for every reabsorbed substance, and it reflects the number of carriers in the renal tubules. When they're saturated, the excess is excreted in the urine. As an example, in diabetics, what you have is a very, very high blood glucose level. So high that all the glucose in the filtrate cannot be put back into the bloodstream. As a result, some of it remains in the urine. The result of that is that it tends to pull water out of the circulatory system and out of the kidney tissue into the bloodstream, and that results in an increase in blood pressure. I'm sorry, strike that. Pulls it out of the kidney and into the filtrate, and that results in an excess in urine volume. Okay, and that's what's called polyuria. Also, you see glycosuria. You see glucose in the urine. You see excessive urination. You also see dehydration. Okay, so that's the net result at the kidney. Okay, so the kidney is dumping a lot of fluid. You're losing a lot of fluid volume. In the proximal convoluted tubule, we see the site of most reabsorption. All nutrients are reabsorbed here, and 65% of the sodium and water, as well as a lot of ions. All the uric acid and about half the urea are reabsorbed here and later secreted back into the filtrate. And this is how we get rid of nitrogen-containing waste. Basically, urea is a chemical compound that's made by combining carbon dioxide to 2 molecules of ammonia. Ammonia is a very toxic compound in the interstitial fluid or in the blood. And so in order to carry it in a more palatable form, we combine it with carbon dioxide and then we excrete that in the urine stream. The nephron loop has a descending limb through which water can leave but solutes cannot. The ascending limb has different properties. Water cannot leave and solutes are actively pumped into the kidney tissue. The thin segment allows passive sodium movement, while the thick segment utilizes a sodium-potassium chloride symporter and a sodium hydrogen ion antiporter, as well as some of this passing through by a paracellular route. The bottom line is in the ascending limb, we pump sodium into the kidney. And as a result, the solute concentration of the tubule fluid begins to drop as the filtrate climbs back up towards the distal convoluted tubule. In the DCT and the collecting duct, we have reabsorption that's regulated by hormones. ADH controls water permeability of the collecting duct, while aldosterone, our sodium retaining hormone, also has an effect on water movement. When aldosterone is secreted, we retain more sodium, and as a result, we retain more water. This increases blood volume, and as a result, increases blood pressure. Atrial natriuretic peptide causes sodium to be secreted, and as a result, this lowers blood volume and blood pressure and increases urine volume. Parathyroid hormone causes calcium to be taken from bone and put into the blood plasma. Okay, this is one of the ways that we control blood calcium levels. Excess secretion of parathyroid hormone results in early onset osteoporosis. Parathyroid hormone also increases reabsorption of calcium from the small intestine. So the net effect is to raise blood calcium levels. Antidiuretic hormone is released by the posterior pituitary, which hangs just under the hypothalamus of the brain via the infundibulum, the little stalk that connects the two. This causes the principal cells of the collecting ducts to insert aquaporins in the apical membranes, and this leads to increased water reabsorption, increased water retention, and as a result, increased blood volume and blood pressure. Aldosterone targets the collecting ducts, specifically the principal cells in the distal convoluted tubule, and it promotes a synthesis of luminal sodium and potassium channels and basolateral sodium-potassium ATPases for sodium reabsorption, and then water follows. Very little sodium leaves the body. Aldosterone absence results in loss of 2% of filtered sodium daily, and this would be incompatible with life. What are its functions? Well, it increases blood pressure and it decreases potassium levels. So, along with ADH, this is one of the ways that we control systemic blood pressure. Atrial natriuretic peptide reduces blood pressure and blood volume. It reduces blood sodium by decreasing blood volume and blood pressure. What happens is, as the sodium is excreted into the filtrate, fluid leaves the interstitial fluid and the blood plasma is lost in the urine stream. As a result, blood pressure and blood volume go down. It's released by the cardiac atrial cells if blood volume or pressure are elevated. Parathyroid hormone acts on the distal convoluted tubule, and this increases calcium reabsorption at the DCT. And so what happens here is that we rescue more calcium out of the filtrate, we put it back into the blood plasma. This further elevates the blood calcium. So this is the third mode of action of PTH that elevates blood calcium. The first being that it steals it from the bones by elevating osteoclast activity. It absorbs more of it from the small intestine, and it also causes more reabsorption in the distal convoluted tubule of the kidney. The bottom line, to increase blood calcium levels. Calcitonin does the reverse. Calcitonin lowers blood calcium levels by increasing osteoblast activity, decreasing osteoclast activity, and as a result, dropping blood calcium. Reabsorption in reverse is what would describe tubular secretion. Secretion is to put components into the filtrate and eliminate them from the body. Reabsorption is to take components out of the filtrate and put them into the bloodstream or the interstitial fluid. Almost all of this occurs in the proximal convoluted tubule. Selected substances such as potassium, protons, and ammonium, as well as creatinine, organic acids, and bases, move from the peritubular capillaries through the tubule cells and into the filtrate. Substances that are synthesized in the tubule cells are also secreted. such as bicarbonate. And this is one of the ways that we control blood pH, by controlling the acidity or the alkalinity of the urine. We keep the blood pH within that nice range between 7.35 and 7.45. Tubular secretion disposes of substances such as drugs that are bound to plasma proteins, and it eliminates undesirable substances that are passively reabsorbed, such as urea and uric acid. It also helps rid the body of excess potassium, and this is the aldosterone effect, while controlling blood pH by altering amounts of protons or bicarbonate in the urine. And so this is a diagram showing you the different phenomena that occur in different regions along the nephron. So there's Bowman's, there's the PCT, there's the descending limb of the loop, the ascending limb of the loop, and there's the DCT, and then finally the collecting duct. And so how do you read this diagram? If we're secreting something, it's going to be a green arrow going into the tubule. If we're reabsorbing something, it's going to be a blue arrow coming out of the tubule. And the box lists what's moving in and what's moving out in different locations along the nephron. Now, with the proximal convoluted, or rather with the distal convoluted tubule and the collecting duct, the amount of reabsorption can vary depending on the hormone influence, such as parathyroid hormone, ADH, or aldosterone. The transporting that goes on in the PCT and in the loop is relatively constant. The most important thing that the loop does is establish the concentration gradient that is used by the collecting duct to finally achieve the final concentration of the urine as it leaves the body. Without this concentration gradient in the kidney proper, which is set up by movement of sodium out of the filtrate and into the kidney, as well as the urea that's already there, we would have no way of driving water movement out of the filtrate and back into the bloodstream and the interstitial fluid as we pass through the collecting duct. Okay, so how do we regulate urine concentration and volume? Well, we need to get our minds wrapped around the concept of osmolality. This is the number of solute particles in a kilogram of water, and it reflects the ability of that particular volume of solute to cause osmosis. It's expressed in milliosmoles. The kidneys maintain the osmolarity of plasma at about 300 milliosmoles by regulating urine concentration and volume. The kidneys regulate this with countercurrent mechanisms, and the countercurrent mechanism simply details the fact that in the nephron, the movement of the filtrate and the movement of the blood that surrounds it are generally in opposite directions. And what that does is it increases the efficiency with which we either secrete compounds into the filtrate or reabsorb compounds out of the filtrate. This occurs when fluid flows in opposite direction in two adjacent segments of the same tube within the hairpin turn. The countercurrent multiplier is the interaction of the filtrate flow in the ascending or descending limbs of the nephron loops of juxtamedullary nephrons, which, as you recall, make up about 15% of all the nephrons in the kidney, but they're important because they are what control the fluid balance, and also, by extension, they are what set up the concentration gradient in the kidney. The countercurrent exchanger is blood flow in the ascending or descending limbs of the vasa recta, and again, this is going to be opposite the direction of flow of the filtrate in the nephron. So what is the role of the countercurrent mechanism? It's to establish and maintain an osmotic gradient from the renal cortex through the medulla. It allows the kidneys to vary the urine concentration as well. And so what you're looking at here is a diagram that shows you how the osmolarity of the filtrate changes as we move from the cortex deep into the medulla and then back up into the cortex again. And so what dictates how this operates? Well, again, what's happening in Bowman's? Bowman's is primarily filtration of pretty much everything, right? dictated by the size of the molecule. If the molecule is below a certain molecular weight, it's going to pass into Bowman's and it's going to be part of the filtrate. In the proximal convoluted tubule, what do we primarily do? We rescue solutes necessary to maintain life. Things such as glucose, amino acids, electrolytes, and a lot of water. Then as we descend down the loop of Henle, what happens here is that these long juxtamedullary loops create the gradient, and they act as countercurrent multipliers. What's happening here as we descend is that water is leaving the filtrate and is entering the kidney tissue, and as a result is also entering the blood that's in the vasa recta. Okay, so we're adding volume to the blood plasma. We're also adding water to the kidney. Well, what's happening to the filtrate at the same time? Well, the osmolarity is going up. This is a graph showing you the osmolarity of the filtrate as we descend down into the kidney. The osmolarity in the filtrate also reflects the osmolarity in the surrounding tissue fluid. Okay, so as we go down the descending limb, the osmolarity goes up, 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 up. Where we get to the bend in the loop, the osmolarity is at its maximum, right around 1,200 milliosmoles per liter. What's happening as we make the turn up is that the tube changes its game. And now pumps inside the ascending limb start to actively move sodium out of the filtrate and into the surrounding tissue. The more concentrated the solute is, the hotter these pumps run. As a result, deep in the medulla, you're going to have more salt pumped out per unit time, more sodium rather, than you are going to have more towards the cortex. Okay? And that's why as we go deep into the medulla, we're very, very salty. The gradient's very, very strong. The gradient gets less strong as we head back up towards the cortex. But it's this last movement of filtrate through the collecting duct which determines the ultimate concentration of the urine. And again, that is controlled by the interaction between this gradient, but also by the permeability of the collecting duct to water proper, which is governed by mostly ADH. So in the descending limb, we have free movement of water. This passes out of the filtrate and into the hyperosmotic medullary interstitial fluid. The osmolarity increases to about 1,200 milliosmoles. In the ascending limb, we're impermeable to water, but permeable to solutes. Sodium and chloride are actively reabsorbed in the thick segment. Some of it is passively reabsorbed in the thin segment, and the filtrate osmolality decreases to about 100 milliosmoles as we ascend towards the cortex. A constant 200 milliosmol difference between the two limbs of the nephron loop and between the ascending limb and the interstitial fluid are what are responsible for this countercurrent multiplier mechanism. The difference is multiplied along the length of the loop to about 900 milliosmoles. In the vasa recta, we preserve the medullary gradient. This prevents rapid removal of salt from the interstitial space. It also helps remove reabsorbed water. Water entering the vasa recta, either from the descending vasa recta or is reabsorbed from the nephron loop and the collecting duct, accounts for the volume of blood at the end of the vasa recta being greater than at the beginning. And that only makes sense, right? Because as we move fluid out of the filtrate into the kidney and into the blood plasma, the volume of blood is going to go up. And so here you're looking at a diagram showing you what's happening in the different parts of the loop. Okay, so in the descending limb, fluid is flowing in the opposite direction through the two adjacent portions of the nephron loop. It's also flowing in the opposite direction of the blood in the vasa recta. As we descend, we lose water and gain osmolarity. Look how concentrated the filtrate is at the bend of the loop, 1,200 milliosmoles, okay, per liter. And then, as we work our way back up through the ascending limb, we actively pump salt out of the filtrate. The osmolarity drops as we ascend all the way back up to the renal cortex. So the countercurrent multiplier depends on three properties of the nephron loop in order to establish this osmotic gradient. The fact that we're moving in opposite directions, and the fact that here we're permeable to water, and here we're impermeable to water and permeable to solutes. Okay, these properties establish a positive feedback cycle that uses the flow of fluid to multiply the power of the salt pumps. So here we have the interstitial fluid osmolality in the filtrate. Water leaves the descending limb. The osmolality goes up as a result of the filtrate losing water to the surrounding tissue. The osmolality of the filtrate entering the ascending limb is very high. As a result, we pump more sodium and chloride out per unit time. The salt is pumped out of the ascending limb, and then we're back to the cortex. Okay? So over and over and over again, we establish this interdependent relationship. And here you can see a diagram showing you what's happening in each part of the nephron. Okay? And what we're diagramming for you here is the change in osmolality as we move through the actual nephron. Okay, so see how we are in the Bowmans, right? We're at 300 milliosmoles, and then as we head through the PCT, we descend now. We're still at 300 milliosmol, and then what happens is that water is beginning to leave the filtrate as we descend deep into the medulla. Okay, now look at what's happening to the osmolality and the osmolarity, basically two measurements that are essentially the same. It begins to climb, right? We lose water, we leave behind solutes, water moves out of the filtrate and into the in the descending limb down its osmotic gradient, and this causes the filtrate to become very, very concentrated. The filtrate reaches its highest concentration at the bend in the loop, and then as we ascend, sodium and chloride are pumped back out. This increases the interstitial fluid osmolality, and then finally we re-achieve now the cortical osmolality in the distal convolute till you're about 100 milliosmoles or so. And now here we can see what's happening in the vasa recta. Okay, this is blood flowing in from the afferent arteriole, and the osmolality is about 300. Now what's happening? Well, what's happening in the accompanying filtrate is that the osmolarity is climbing as we descend in the medulla. Now look what's happening to the osmolarity of blood. It's beginning to climb as well. Okay, why is that? Well, because the capillaries are permeable to water and solutes. So what's happening now is that sodium chloride is entering, all right, and water is leaving. Okay, the countercurrent flow of the fluid through the two adjacent parallel sections of the vasa recta is responsible for this exchange. Okay, now as we reach the bend down here in the loop, we're still in the blood vessels here. Look what's happening to the osmolarity. It's basically assuming the osmolarity of the surrounding fluid. So 1200 milliosmoles here, and then we begin to drop until we get all the way back up to what's eventually going to enter the interlobular veins, and we're around 300 milliosmoles once we're back into the cortex. Okay, so basically what's happening in the blood is that we're mimicking what's happening in the surrounding gradient that's again established primarily by those long loops of Henle. So the entire length of the vasa recta is permeable to water and solutes due to the countercurrent exchange between each section of the vasa recta and its surrounding interstitial fluid. The blood within the vasa recta remains nearly isotonic to the surrounding fluid. As a result, the vasa recta do not undo the osmotic gradient as they remove reabsorbed water and solutes. And now we can take a peek at what happens in the collecting duct. Well, what happens in the collecting duct is, provided it's reasonably permeable to water, the osmolality of the filtrate is going to increase as we dive back down through the medulla. Okay, so look how concentrated the urine gets before it eventually leaves the collecting duct, enters the minor calyx, enters the major calyx, enters the renal pelvis, and ultimately goes from the ureter to the bladder to the urethra, and that's the end of the trip. Okay, so how do we form dilute or concentrated urine? The osmotic gradient used to raise urine concentration is greater than 300 milliosmoles in order to conserve water. If we have overhydration, we make large volumes of dilute urine. ADH production is reduced, and the urine has a milliosmolarity of about 100. If aldosterone is present, additional ions are removed, and we can get down to 50 milliosmoles. At this point, the urine almost looks clear. In dehydration, we have a small amount of concentrated urine. Why is this? Well, we want to hold on to as much water as we possibly can. ADH is released, and the urine is very concentrated, up to 1200 milliosmolar, and will have usually a dark yellow to almost orange color. In severe dehydration, 99% of the water is reabsorbed from the urine before it leaves the body. And this is just showing you how these phenomena change depending on whether or not we're overhydrated or whether we're dehydrated. The bottom line, if we're overhydrated, dilute urine is created. If we're dehydrated, very concentrated urine is created. The bottom line, we hold on to water when we need to, and we eliminate excess fluid when we consume too much. Now, you can overload the system if you do something ridiculous, like, I don't know, drink 5 liters of distilled water in under 2 minutes. Okay, even the kidneys won't be able to compensate rapidly enough to restore fluid and electrolyte balance. You'll die of cerebral edema. Same can be said for severe dehydration. The kidneys can hold on to a lot of water, but they can't create water out of nothing. And eventually what will happen is that you'll lose enough fluid volume, even if you're making concentrated urine, that you'll dehydrate to the point where you'll die. Urea helps form the medullary gradient. It enters the filtrate in the ascending thin limb of the nephron loop by facilitated diffusion. The cortical collecting duct reabsorbs water and leaves urea behind. In the deep medullary region, a highly concentrated solution of urea leads to the interstitial fluid of the medulla, which leads back to the ascending thin limb, which results in a high osmolality deep in the medulla. Okay, so the bottom line again is that it contributes to the osmotic gradient. The osmotic gradient increases the deeper in the medulla you go. Diuretics are chemicals that enhance urinary output. Examples include ADH inhibitors such as alcohol. Sodium reabsorption inhibitors and the resultant water reabsorption can be triggered by caffeine or drugs for hypertension or drugs for edema. Loop diuretics inhibit the medullary gradient formation, while osmotic diuretics are substances that are not reabsorbed so the water remains in the urine. Examples would be the high glucose of a diabetic patient, because you overload the ability of those pumps to put the glucose back into the bloodstream. So when the glucose stays in the filtrate, it draws water to it and it increases the volume of the urine. How do we evaluate kidney function? Well, we look at the output. We examine the urine for signs of disease and assess renal function using both blood and urine examinations. Basically, do a chemistry profile. Renal clearance is the volume of plasma that the kidneys clear of a particular substance in a period of time. Renal clearance tests are used to determine the glomerular filtration rate. They're also used to detect glomerular damage and to follow the progress of renal disease. We can use a formula to relate renal clearance to known factors. Clearance is equal to the concentration of a substance in the urine times the flow rate of the urine formation divided by the concentration of the same substance in the plasma. Okay. Inulin is a plant polysaccharide that's neither secreted or reabsorbed. It's freely filtered. Its renal clearance has a GFR of about 125 milliliters per minute. If it's less than that, the substance is going to be reabsorbed If it's equal to that, the substance is completely reabsorbed or not filtered. If it's equal to 125 milliliters a minute, there's no net reabsorption or secretion, and that's what you would expect with an inulin dose. If the concentration is greater than 125 milliliters per minute, the substance is secreted, and this occurs in most drug metabolites. Chronic renal disease is characterized as a glomerular filtration rate of less than 60 milliliters per minute for 3 months. Examples include individuals in diabetes mellitus or hypertensive states. Renal failure is classified as a glomerular filtration rate of less than 15 milliliters a minute, and this can cause uremia, where ionic and hormonal imbalances result from the buildup of nitrogenous wastes in the blood. This results in metabolic abnormalities and toxic molecule accumulation. It's generally treated with hemodialysis or with transplant. If we look at urine, it should be transparent. It should be clear. If it's cloudy, this can indicate a urinary tract infection. It's generally pale to deep yellow from the presence of urochrome, which is a breakdown product of hemoglobin. The pigment from hemoglobin results from its breakdown. In more concentrated urine, it's going to have a deeper color. It's going to be dark yellow to orange. Abnormal colors include pink, brown, or smoky color. This can result from food ingestion, the presence of bile pigments, the presence of blood, or the presence of certain drugs in the urine. It should be slightly aromatic when fresh and develops ammonia upon standing as a result of the bacteria breaking down the urea in the urine. It can be altered by some drugs and vegetables. Asparagus is an example. It should be slightly acidic with a pH range of anywhere from 4.5 to 8. Notice the wide pH range here. And this is because one of the ways that the body maintains blood pH within that nice tight limit is to change the pH of the urine as your dietary intake changes in terms of the acidity or basicity of the foods and drink you consume. An acidic diet, such as protein or whole wheat, will decrease the pH of urine, while an alkaline diet or prolonged vomiting or UTIs will tend to elevate the pH. The specific gravity is a little bit over 1. It depends, again, on solute concentration. Specific gravity is just the density of the urine compared to the density of water. Water is assigned a specific gravity of 1. So if you were to take a drop of urine and place it in a container of water and watch it very carefully, you would generally see the drop of urine sink because of its increased density. There's ways to do this too. You can use refractometers, and you can also use a specific gravity detector, which looks a little bit like a buoy with a level in it, and depending on how deep the buoy sinks in the urine, it'll give you a specific gravity reading. That's one of the old ways to do it. Urine is about 95% water, 5% solutes on average. It contains nitrogenous waste such as urea, which comes primarily from amino acid breakdown. And this is the largest solute component. It also contains uric acid from nucleic acid breakdown, and creatinine, which is a metabolite of creatine phosphate. Other normal solutes include sodium, potassium, phosphate, and sulfate, as well as calcium, magnesium, and bicarbonate. Abnormally high concentrations of any of these constituents or abnormal components in the urine, such as blood proteins, white blood cells, bile pigment, or bacteria, can indicate pathology, right? A problem with the urinary tract or with kidneys. So once we go through all the trouble of making the urine, how do we get it out? Well, we have to move the urine from the kidneys to the bladder, and that's done at about lumbar 2 as a continuation of the renal pelvis, into which the major calyces drain. Okay, or the minor calyces catch the urine as it comes out of the renal papilla, which is the tip of the renal pyramid. Uh, the minor calyces dump the urine into the major calyces. The major calyces dump the urine into the renal pelvis, and from there we enter the ureter. Um, the kidneys, remember, are retroperitoneal. They enter the base of the bladder through the posterior wall of the bladder. As the bladder pressure increases, the distal ends of the ureters will close, and this normally prevents backflow of urine. The ureters have three layers of wall from inside to the outside. The mucosa is made up of a transitional epithelium, meaning it has the ability to stretch. The middle or muscularis layer has a smooth set of smooth muscle sheets, so it's capable of contracting in response to stretching. And it also helps propel the urine into the bladder. And the adventitia is an outer fibrous connective tissue layer that anchors it to the body wall. And this is just a look at the different layers of the ureter. It's just a transverse section through the ureter. The lumen is the white area on the inside here. The mucosa is this purple staining layer, and it's made up of a transitional epithelium and a lamina propria. Okay, the lamina propria is the underlying connective tissue layer. The transitional epithelium is the actual tissue that has the ability to stretch because of the presence of elastic proteins in the extracellular matrix of this tissue. The muscularis has a longitudinal and a circular layer. This is a very similar arrangement to what you might find in the esophagus or in the small intestine. Okay, same basic layout. And then you've got an outer layer connective tissue adventitia out here. Sometimes we can have electrolyte or fluid imbalances. This can result in pathological conditions in the kidney. Renal calculi are minerals that can deposit in the renal pelvis or lodge in the ureter. They're often crystallized calcium, magnesium, or uric acid salts. The type depends on the nature of the imbalance. The large stones can block the ureter. This results in pressure and severe pain. And this is referred pain. This is not pain where you'd expect it. You'd expect the pain to be where the kidneys are located, right? Which is along the posterior body wall just under the diaphragm. But that's not actually where you feel the pain. You actually feel the pain through the pelvic area. It feels a little bit like severe constipation. This can be due to chronic bacterial infections, urine retention, elevated calcium in the blood, or elevated pH in the urine. One of the treatments is shockwave lithotripsy, where we use sound waves to try and smash the stones. This helps them become easier to pass. Another way is to ingest large quantities of water in an effort to dilate the ureter and pass the stone more easily. This can also help in dissolving the stone. The bladder is a muscular sac for temporary storage of urine before it's eliminated. It is retroperitoneal, so it's behind the peritoneal membrane. It's on the pelvic floor, posterior to the pubic symphysis. And for you gals in the class, it's right underneath the uterus. The uterus actually leans forward over the urinary bladder anteriorly. In males, the prostate gland lies underneath the bladder, and it actually is the first area through which the male urethra passes. In females, it's anterior to the vagina and to the uterus. There are openings for the ureters and the urethra in the bladder. This is called the trigony. This is a smooth triangular area that's outlined by these openings. Infections tend to persist in this region. There are also layers to the bladder wall. The mucosa is made again of transitional epithelia, so it has the ability to stretch. There's also a detrusor muscle, which is basically a three-layered smooth muscle that helps propel the urine out of the bladder and into the urethra. And then the outer surface is a fibrous adventitia, which is peritoneum on the superior surface only. The urinary bladder collapses when it's empty, and wrinkles called rugae appear in the inner surface. It expands and rises superiorly during filling without a significant change in the internal pressure. A full bladder, 12 centimeters long, holds about half a liter of fluid. It can hold about twice that if necessary, but it can burst if it becomes over-distended. This is just a peek at your urinary bladder where it's located. Notice it's well protected by the pelvic girdle. Okay. This is the renal pelvis. There's a kidney. Okay. Kidney there. Renal pelvis. Ureter. There's your left ureter. There's your right ureter coming into the back of the urinary bladder. Urethra would be down here. Okay. Here you can see the regions of the urethra in the male. Okay. Here are the ureters coming in the back. There's the bladder. There's the trigony right there. Okay. This is looking from the top down. Okay. This is superior view. There's the prostate gland. Prostate gland lies directly under the bladder and is directly posterior to the bulb of the penis, that part of the penis that's closest to the body wall. This is the prostatic urethra. This is the portion of the urethra in men that becomes occluded in prostatitis or prostate cancer. This is the membranous urethra, this little length of tubing that connects the prostatic urethra to the penile urethra, which runs through the shaft and the glands of the penis. It's surrounded by a corpus spongiosum, which is a vascularized erectile tissue, and on its superior aspect, it's covered by the corpora cavernosa, okay, that diverge like a Y on top of either side of the urethra. When the male has an erection, what happens is the blood flow into these erectile tissues is greater than the blood flow out. The result is an erect organ. And this is a look at the situation in the female. Again, the ureters enter in the back. This is the bladder. Here's the trigony again. Notice that the urethra is much shorter in the female than in the male. Okay, this is the external urethral orifice. There's the urogenital diaphragm, and there's the external urethral sphincter. This is the part that you control. The internal urethral sphincter is under involuntary control. Okay, this is smooth muscle. To look at the man has these structures as well. Okay, there is the external urethral sphincter in the male, and the internal urethral sphincter is up here on the other side of the prostate gland. Okay, so you can see these structures also in the male. Incidentally, those are the bulbourethral glands, prostate gland. Again, they contribute components to the semen. Okay, so the urethra is a muscular tube that drains the urinary bladder. It has an epithelial lining that's made up mostly of pseudostratified columnar epithelia, except for the transitional epithelia that's near the bladder, which has the ability again to stretch. There's stratified squamous epithelia near the urethral orifice. There are sphincters that control the passage of urine through the urethra. The internal urethral sphincter is smooth muscle. It's under involuntary control and it's situated at the bladder-urethra junction. It contracts in order to open, while the external urethral sphincter is skeletal muscle surrounding the urethra as it passes through the pelvic floor. The female urethra is 3 to 4 centimeters long and it's tightly bound to the anterior vaginal wall, and the exterior urethral orifice is anterior to the vaginal opening and posterior to the clitoris. And again, this is just a revisitation of the diagram that we saw earlier. Okay, so you can again see the openings for the ureters as they pass into the bladder. You can also see here the rugae, the folds, when the bladder is relaxed. There is the peritoneal layer. There is the internal urethral and external urethral sphincter. Okay, here, external urethral sphincter, internal. Okay, and there's the external orifice. The male urethra carries both semen and urine. It has 3 regions: the prostatic urethra, it's contained in the prostate gland; the intermediate part of the urethra, known as the membranous urethra, which passes through the urogenital diaphragm from the prostate to the beginning of the penis; and the spongy or penile urethra that passes through the penis and opens via the external urethral orifice. And this is, again, just another peek at what we already looked at. Okay, so again, you can see the 3 regions of the male urethra: prostatic, membranous, and penile, okay, or spongy. The reason it's called the spongy urethra as opposed to the penile urethra in some textbooks is because it goes through the corpus spongiosum, that erectile tissue that encloses this chamber. Okay, micturition is basically the act of urination. There are three simultaneous events that have to happen. We have to have a contraction of the detrusor muscle to squeeze the bladder. We have to open the internal urethral sphincter. This occurs as a result of input by the autonomic nervous system. And we have to open the external urethral sphincter by voluntary control. Okay, the somatic nervous system comes in there. Reflexive urination is what's seen in infants. Distension of the bladder activates the stretch receptors, and excitation of the parasympathetic neurons in the reflex center in the sacral region of the spinal cord lead to contraction of the detrusor muscle and the opening of the internal sphincter. Inhibition of somatic pathways to the external sphincter allow it to relax, resulting in its opening, and the urine flow begins. Pontine control centers mature between ages 2 and 3, and these storage centers can inhibit urination by inhibiting parasympathetic pathways and exciting sympathetic and somatic efferent pathways. Pontine micturition centers promote micturition by exciting parasympathetic pathways and inhibiting sympathetic and somatic efferent pathways. And this is just a flow chart showing you how these different pathways interact. This is 2521 in your book. Take a peek at this. I won't go through this in a lot of detail, but essentially the bottom line is that in infants we don't have the higher brain centers developed yet to control the micturition reflex. As a result, involuntary urination is always a danger. Incontinence usually comes as a result of weakened pelvic muscles. In stress incontinence, we have increased intra-abdominal pressure that can force the urine through the external sphincter, even though we attempt to voluntarily keep it closed. In overflow incontinence, the urine dribbles when the bladder overfills. Urinary retention is when the bladder is unable to expel the urine. This is common after anesthesia or when the prostate hypertrophies. When it hypertrophies, it can occlude the prostatic urethra. As a result, the urine cannot move into the membranous or penile urethra. The treatment is generally catheterization, which is a tube that's inserted through the urethra into the bladder in order to drain it. There's three sets of embryonic kidneys that form in succession during human development. The pronephros degenerate, but the pronephric ducts persist. The mesonephric ducts claim this duct and become the mesonephric ducts, and the metanephros develop by the 5th week, and this develops into the adult kidney and begins to ascend inside the body cavity. And so here you can see an example of how these ducts work. Okay, so to orient you, this is going to be your head. This is going to be your eye. Okay, this is going to be your navel. This is eventually going to be your coccyx. Okay, right at this point, it's your tail. So these are the degenerating pronephros. This is at week 5 during development. And this is a piece of tissue called the urogenital ridge. The mesonephros begin to develop along this ridge. Okay. The mesonephric duct is the channel through which they move. Okay, so you can see here the mesonephros, the pronephros, the mesonephros, and then what's going to happen is that this tissue is actually going to move inside the embryo. This is called morphogenetic movement. This is done primarily as a result of chemical cues that are given off by developing tissue. This is the ureteric bud. Okay? The cloaca is actually a primitive structure. We see this, for instance, in reptiles and birds, and it's a common opening through which fecal material and urine pass. So this is another one of those primitive structures that appears and disappears during human development. As we move on, this is week 6, the pronephros are beginning to degenerate. Mesonephros are persisting, right? And the metanephros are starting to elaborate down here, okay? So this is beginning to grow, and it's beginning to move in a rostral direction, superiorly, and also posteriorly. Eventually what's going to happen is it's going to ascend all the way up to the area of the body wall that's just underneath the diaphragm. The metanephros develops as the ureteric buds that include the mesoderm of the urogenital ridge, and this forms the nephrons. The distal ends of the ureteric buds form renal pelvis, calyces, and collecting ducts, and the proximal ends become the ureters. The kidneys excrete urine into the amniotic fluid by the 3rd month. The cloaca then subdivides into the rectum, the anal canal, and the urogenital sinus. So here we are at week 7. Okay, so what's happened? Well, the promesonephros are almost completely gone now, right? You can still see a lot of these mesonephros, but they're decreasing in size, okay? But they're still around. This is eventually going to be the gonad in the female. This would be the ovary. In the male, this would be the testes. Here you can see what's eventually going to be the rectum. This is the urogenital sinus, okay? And this would be eventually the developing urinary bladder, And you can see what is eventually going to be the kidney, and this is what's eventually going to be the ureter, okay? All right, so what happens next? So as we move from week 7 to week 8, what do we see? Well, the kidney begins to elaborate and ascend, okay? Mesonephros are still there, but they're beginning to shrink in size. You can see now the ureter full-fledged. There's the renal pelvis. There's the ureter. Okay. You can see the rectum and the anus there. This is eventually going to be the urethra, and there is the urinary bladder. Okay. This eventually is going to be the intestine. You can see it beginning to develop here. Okay. Moving right down into the anus. What happens next? Well, eventually what happens is the kidney fully elaborates, the mesonephros disappear, and the ureter and the bladder begin to elaborate. There's 3 common congenital abnormalities that can affect this developing urogenital system. In horseshoe kidney, the 2 kidneys fuse across the midline and form a single U-shaped kidney. Usually this is asymptomatic. In a hypospatus, the urethral orifice is on the ventral surface of the penis. So it's on the underside of the penis instead of at the anterior aspect of the glands. This can be corrected surgically at 12 months. Polycystic kidney disease results in many fluid-filled cysts that interfere with kidney function. This can be a genetic disorder. There's an autosomal dominant form, which is less severe but more common. And then there's an autosomal recessive form that's very severe and results in kidney failure. The cause is unknown, but it involves a defect in certain signaling proteins. In the recessive form, you need 2 bad copies of the gene in order for the disease to show up. In the autosomal dominant form, you only need 1 bad copy. So this is one of the things that you can screen for using amniocentesis. If you know that polycystic kidney disease runs in your family, you can check the cells in the amnion by sampling them prior to the birth of the child, and then performing PCR on the genetic material that you isolate from these cells, and checking the DNA sequence for the genes that are suspected to be defective. And then you'll know conclusively whether or not the child is going to have the disease or carry the gene, depending on which form of the disease that happens to run in your family. Frequent micturition in infants is usually due to small bladders and less concentrated urine. Incontinence in normal infants has to do with the control of the voluntary urethral sphincter and develops with the nervous system as it develops. E. coli account for about 80% of all UTIs, and again that's because of the relative close proximity of the anus to the urethra, especially in women. Untreated childhood strep infections can also cause long-term renal damage, and STDs can also inflame the urinary tract. Most elderly people have abnormal kidneys histologically. They shrink, the nephrons decrease in size and number, and the tubule cells are less efficient at doing their job. Your GFR drops to half that of a young adult by the time you're 80. And there's also nephrosclerosis and arteriosclerosis of the renal artery that affects the filtration function. The bladder begins to shrink, and we lose bladder tone, and we can also see nocturia and incontinence. Nocturia is urination at night when you're asleep, and incontinence is an inability to hold the urine flow, and that can result from a variety of reasons, among them, in the elderly, a loss of muscle tone. Okay, that brings us to the end of this portion of the text. I will see everybody Saturday. Make sure and review the material in your textbook. And any questions that you have that are still confusing to you, get in touch with me via email or by cell phone. See you soon.